Hi everyone, welcome to my YouTube channel, and today we will be looking at one of the most classical tenets of genetics, which is your Mendelian genetics. But before that, don't forget to like and subscribe my YouTube channel. Let's start. This is Gregor Johann Mendel, and he's one of considered as the father of genetics. In 1866, he put forward the major postulates of transmission genetics, which started with his experimentations with the garden pea plants. And his major breakthrough was a big idea that was being used in a different subdivision of genetics, which we call as your transmission genetics. So this presentation we'll be looking at inheritance, okay, which is governed by the information stored in discrete unit factors, which we call them genes. And later we will be talking about what these unit factors are. Genes are also transmitted from generation to generation. On the vehicles, we call them chromosomes. And chromosomes, as you know, are found on your cells, okay, especially inside the nucleus. Chromosomes, which are existing in pairs in diploid organisms, are providing us a basis for biparental inheritance, the one coming from your mom and the one coming from your dad. Next would be during gamete formation, these chromosomes are distributed according to the postulates, which is first described by Mendel based on his 19th century research with his garden peas. And his postulates will prescribe that homologous chromosomes will segregate from one another and they assort independently with other segregating homologs during the gamete formation. And also we will be looking at genetic ratios, which are being expressed as probabilities which are subject to chance deviation and may be evaluated statistically. And lastly, the analysis of pedigrees. Okay, these pedigree charts allows prediction concerning the genetic nature of certain human traits, and that includes diseases for humans. Now, we'll be looking at how Mendel used a model experimental approach to study the patterns of inheritance. And in this next slide, you can see the characters or the phenotypes of these pea plants coming from seed shape, seed color, pod shape, pod color, flower position, and the stem height. So let's say for example, your seed shape, you have round or wrinkled. Your seed color is either yellow or green. Your pod shape is either full or constricted. Your pod color should like green or yellow. Flower color, it's either violet or white. Flower position is either on eggshell, which is on the nodes of your stem, or it's either terminal, which is found on the ends of the stem. Lastly, your stem height is either tall or dwarf. So you can see here in this diagram the 7 pairs of what we call as contrasting traits. Okay, and these contrasting traits are result of Mendel's 7 monohybrid crosses of the garden pea, or shall we say the Pisum sativum. And each case, okay, in each of these character and contrasting traits, you'll see that from the pollen, okay, derived from the plants which exhibited one trait, let's say one round or one for wrinkled, is able to fertilize, or he used it to fertilize the ova of plants exhibiting the other trait. Okay, and in the F1 generation, or shall we say the filial 1 results, okay, one of the 2 traits was exhibited by all the plants. It's either all round or all yellow, all full, all green, all violet, all eggshell, and all tall. And the contrasting trait reappeared in your F2 or filial 2 results, which approximately 1/4, okay, of your F2 plant. So we will be also talking about what these ratio are in the next slides. So now your monohybrid cross will reveal how one trait is being transmitted from generation to generation. Monohybrid crosses, okay, is one of the most simple cross which Mendel used in his experimentation. These monohybrid crosses is involving only one pair of contrasting traits. It's either yellow or green. It's either dwarf or tall. And we will examine the first generation of offspring of such a cross, and then we consider the offspring of selfing. Okay, this selfing is also known as your self-fertilization. This self-fertilization or selfing is coming from the first generation. So these plants like tall, pure tall, they self-fertilize, or pure yellow and pure yellow. Okay, so these are also for monohybrid crosses. The original parents will constitute the P1, so we call them the parental generation. Okay, their offspring are what we call as the F1 or the first filial generation, and the individuals resulting from the self F1 generation are the F2. Okay, F2 are referred to as your second filial generation, and so on. And Mendel's first three postulates will be given into this particular slide. The first one is unit factors in pairs. Okay, unit factors in pairs. That is a very Latinized term or very old term that Mendel is using, because these unit factors are genetic characters which are controlled by unit factors that exist in pairs in individual organisms. In monohybrid cross involving tall and dwarf stem, a specific unit factor exists for each trait. So these postulates, or the first three postulates, are very consistent. Okay, using his consistency on his experimentation, Mendel was able to derive these first three postulates. And yeah, the first one will tell us unit factors. And unit factor is existing, okay, coming from one from the maternal strand and one from the paternal strand. So those are unit factors. Later, we will know what is the unit factor that evolved in the latter. Next will be your dominance and recessiveness. Dominance and recessiveness. When two unlike unit factors responsible for a single character are present in a single individual, one unit factor is dominant to the other, which is said to be recessive. So there is what we call as the terms that we constantly use in Mendelian genetics, which is the dominant and recessive, and they are used to designate traits. Okay, in this case, the tall stems are said to be dominant over the recessive dwarf stems. Your yellow color or pod color is dominant over the green pod color, and so on. The third postulate would be segregation, and it explains that during the formation of gametes, the paired unit factors separate, so they tend to be like segregating with each other, and that segregation is in random fashion. Okay, and each gamete receives one or the other with equal likelihood. And let's say, for example, an individual is containing a pair of these unit factors. Let's say both are specific for tall. Then all of its gametes will receive one of that same kind of unit factor. In this case, like tall. So one offspring will get the tall trait, and the other one will be getting the other trait. If an individual contains unlike unit factors, let's say for example, tall and dwarf, okay, each gamete, okay, each either the sperm or the egg has a 50% of probability of receiving either the tall or the dwarf unit factors. And when filial 1 plants form gametes, the postulate of segregation demands that each gamete randomly again receives either the tall or the dwarf unit factor. Following the random fertilization events during the F1 selfing, 4 F2 combinations will result with equal frequency. So first is your tall or tall, second, tall or dwarf, third, dwarf or tall, and fourthly, dwarf or dwarf. So this tall or tall and dwarf and their combinations, okay, will clearly result in tall and dwarf plants respectively. And according to the postulate of recessiveness and dominance, combinations 2 and 3 will therefore yield tall plants. Okay? Therefore, okay, after the F1, the F2 is predicted to consist, okay, of the 3/4 tall and 1/4 dwarf, or a ratio of 3:1. Again, this is approximately what Mendel is believing when he observed in his cross of like between tall and dwarf plants. A very similar pattern was observed in each of the other monohybrid crosses. So it's between it's either one trait, like tall or dwarf, yellow or green, okay, round or wrinkled. So it includes only one trait. And some terms that we're constantly using in this presentation, we'll be looking at some modern genetic terminologies. Okay, so the first one is traits, such as your tall or dwarf, or physical expressions of the information contained in the unit factors. And this physical expression of a trait is what we call as the phenotype of your individual. What is being observed, what can be perceived by our senses. It could either be physical, morphological, physiological, okay? It could either be behavioral. These are all phenotypes. Mendel's unit factors will represent units of inheritance called genes by modern geneticists. So that the term unit factors, which is Mendel is using, was changed into the genes that are being used by geneticists when they discovered that these genes tend to be passing on, okay, the information from the parents to the offsprings. And for any given character, such as plant height, the phenotype is determined by alternative forms of a single gene, and we call them alleles. And also, when we consider alleles, which were written in pairs to represent the two unit factors that are present in an individual, let's say for instance these letter symbols, the uppercase D and then uppercase D and lowercase d, or both lowercase d, this resulting symbol is, we call them the genotypes. Again, phenotypes or the physical attributes, genotypes are the symbols which are representing in an individual. And when these both alleles are the same, it's either uppercase D or lowercase d, the said individual is homozygous for the trait. And, or let's say we call them homozygote. Okay, homo, same, zygote, the resulting fertilized egg and fertilized egg. And when the alleles are different, like the big or uppercase D and smallcase d, we use the terms heterozygous or heterozygote. And on the next diagram, this is a monohybrid cross between the tall, okay, which we represent as the uppercase D, and dwarf, which is the lowercase d of pea plants. Individuals are shown in rectangles and gametes are shown in circles. So this is your phenotype, which is it will always be, again, unit factors, it will always be 2. This is your tall and this is your dwarf when they are producing gametes, okay, the gamete formation, they will produce a single, okay, genotype big D, uppercase D and lowercase d, and that contributes to the F1 generation. When they fertilized, okay, all of them are tall, okay, and when we self-fertilize or selfing these tall individuals, okay, cross them, and we got these gametes for the F1. Then we now have your F2 generation, and when you get these gametes, then test cross them. We will try to look what are these crosses, and then we are now getting a genotype of this. In pink, we have here a homozygous, heterozygous, and a homozygous recessive traits. Okay, now their phenotypes will tell us three tall and one dwarf organisms, and these are the designation: homozygous, hetero, and homo. So this is technically the we're looking at monohybrid crosses. Okay, and very importantly, there are some techniques or procedure that we are using in genetics that will somehow help you, okay, in looking at and constructing a diagram. So this person is Reginald Punnett, okay, devised an approach, we call them the Punnett square. So the genotypes and phenotypes which are resulting from combining the gametes during your fertilization can be easily visualized during the device, which was again formulated by Reginald Punnett, which is the diagram, we call it the Punnett square. So in this next diagram, you'll see a Punnett square generating the filial 2 generation of your F1 and F1 cross. So this is your F1 cross, all tall organisms, and then we try to produce the gamete formation during the F1 generation, and all of these are gonna get we are getting the uppercase D and lowercase d. So why when we're done with the gamete formation, then now we are trying to set up a Punnett square. So simply put them on the y-axis and the x-axis and draw a table. Okay, so fill out the squares which will represent fertilization process. So just simply cross them. Okay, these are tall, these are tall, these are tall, and this is dwarf. And same thing with what we had in the earlier slides. We got the F2 results, resulting in a 3/4 which is tall and 1/4 which is dwarf. And when we try to convert that one using this ratio, we get a 1:2:1, or generally when we base on the phenotypes or the phenotypic ratio, we're getting 3:1. This is your genotypic ratio. This is your phenotypic ratio. Now, we are presenting you with the test cross. Okay, this is only for a single character. So tall plants which is produced in the F2 generation are predicted to have either the uppercase D, both of homozygous D, or the heterozygous D genotypes. And you might ask if there is a way to distinguish the genotype. Okay, and Mendel was, very curious about it, and he devised a rather simple method that is still used today to discover the genotype of plants and animals, and we call that as your test cross. Now we got a few examples of a test cross coming from a single character. Now first, in letter A, you got a tall parent which is homozygous, and next it will be in letter B a tall parent which is a heterozygous, and they are paired together with a homozygous dwarf parent. Now in the F1, you get these big D for the letter A and small d for the pair. Then the offsprings are all tall. Now, since we are getting a heterozygous tall, and it has either two 50-50 possible gametes, it's either the big D or the small d, and we cross them with the small d, now we're getting an F1 result of it's either half are tall or heterozygous and half were dwarf. So these are the possible outcomes of your test cross. Now, this is a good example or a good activity just to begin with our discussion with Mendelian genetics. So these are pigeons. Okay, pigeons may exhibit a checkered or plain color pattern. So checkered or plain, those are the phenotypes. In a series of controlled matings, the following data were obtained. In your P1 cross, okay, parental cross, we have here a checkered and checkered, and this F1 progeny will give you a 36 individuals which are checkered and 0 plain. For checkered and plain, we got 38 checkered and 0 plain. And lastly, when you combine plain and plain, now we get 0 checkered and 35 plain. And here, okay, we got the F1 offspring, okay, which are selectively mated with the following results. So all of these are coming from the filial 1 generation, because the P1 cross, which give rise to each F1 pigeon, is indicated in parentheses. So in your, this is your checkered and plain, checkered and plain, checkered and checkered, checkered and checkered. So we have here the A and B, ABC. Okay, so these are the following F2 progeny. We have 34, 0, 17, 14, 28, 9, and 39, 0. So the question that you need to answer is, how are the checkered and plain patterns inherited? Okay, and select and assign symbols for the genes involved and determine the genotypes of the parents and also of the offspring in each cross. So generally, this problem will ask you to analyze the data produced from several crosses involving pigeons, okay, and to determine the mode of inheritance and the genotypes of the parents and offspring in a number of instances. And the key to its solution, I'm giving you a clue, okay, is to first determine whether or not this is a monohybrid cross. Okay, to do so, you need to convert the data to ratios that are characteristic of Mendelian crosses. In this case of this problem, ask first whether any of the F2 ratios match Mendel's 3:1 monohybrid ratio. That's the basis. Okay, if so, then the second step is to determine which trait is either dominant and which trait is recessive. So I'm gonna leave you on that activity, and we'll try to move on to the next part of this lesson. Now we're trying to look at Mendel's dihybrid cross, which are generated in a unique F2 ratio. Now we're looking at the extension, okay, natural extension of the monohybrid cross. And Mendel also designed experiments in which he examined two characters simultaneously. And such a cross involving two pairs of such different or contrasting traits is called Dihybrid Cross or two-factor cross. So this is generating two crosses. P1 cross will be yellow and round, green and wrinkled, yellow round, green and wrinkled. Next cross will be yellow and wrinkled and green and round. So we have two different traits that are present here. Now we will try to cross these parents, and now we're getting all yellow and round. We're done with all of the F1s are being self-fertilized. We fertilize them, and we have results: yellow and round, yellow and round. Okay. Now in your F2 generation, we got these results. On this particular results, we get 4 different phenotypes coming from the original parental crosses. Okay, 9 yellow and round. That's because there are 16 individuals produced in this cross. 3 yellow and wrinkled, 3 green and round, and 1 green and wrinkled. This is the least phenotype we're seeing here. Okay, so using the dihybrid crosses used by Mendel, he was able to derive at his fourth postulate, and that is independent assortment. it explains to us that during the gamete formation, okay, segregating pairs of unit factors assort independently of each other. Okay, random. These are segregating independently. They're looking for um, a certain gamete, okay, that is not driven by any molecule or any machinery. And also, this postulate will stipulate that segregation of any pair of unit factors occurs like independently of all others. There's no again internal mechanism that acts upon this, and as a result of this random segregation, each gamete receives one member of every pair, okay, of these what we call as the unit factors or the genes. And for one pair, whichever unit factor is received, it does not influence the outcome of segregation of any other pair. So in general, okay, this postulate of independent assortment will tell us that all possible like combinations of gametes should be formed in equal frequency. They should be placed together like in a diploid organism, like in humans. We should be getting all the 23 pairs of these chromosomes for a normal individual. Like we have the 22 pairs of autosomes in one pair of your sex-linked chromosomes, either a male or female. Now this is a diagram of a computation of the combined probabilities of each of the F2 phenotype for like two independently inherited characters. Okay, so the probability of each plant being yellow or green is independent of it being round or wrinkled. So for this F1 generation, we got yellow round and yellow round from the previous Dihybrid Cross. So of all the offspring, 3/4 are yellow, 1/4 are green. How many of them are round or wrinkled? We have got 3/4 are round and 1/4 are wrinkled. For the greens, 3/4 also are round and 1/4 are wrinkled. So now we combine them, these probabilities, by simply multiplying it. Okay, 3 times 3, it's 9, and 4 times 4, it's 16. So these are the particular phenotypes and genotypic ratios that we got from this F1 generation of these dihybrid cross. And this diagram will be an analysis of a dihybrid cross, which is shown in this figure, okay, showing you that this F1 heterozygous plants, like yellow and round, okay, which is self-fertilized or has been selfing and producing this F2 generation. So before we got this F2 generation from the F1 cross, since they are both yellow and round heterozygotes, we got these gametes, okay, 4 gametes from the x-axis and 4 gametes from the y-axis. Now we are looking at the genotypic ratio and the phenotypic ratio of these individuals, and you notice that there is something in those unit factors or genes that are acting upon in which trait is either dominant or recessive. So when you say the dominance of these yellow and round traits, it will manifest in those particular events that are done in the dihybrid cross. So in Mendel's fourth postulate, okay, every filial 1 to filial 1 fertilization event, each zygote again has an equal probability of receiving one of the 4 combinations from each parent. And if many offsprings are produced, 9/16 are yellow round, have yellow round seeds, 3/16 have yellow wrinkled seeds, 3/16 have green and round seeds, and 1/16 have green and wrinkled seeds. And that's yielding the designated Mendel's 9:3:3:1 dihybrid ratio. Okay, very ideal. This is very ideal ratio, which is based solely on the probability events, which is involving segregation, independent assortment, and random fertilization. And because this deviation is due to strictly chances, particularly if small numbers of offspring are produced, actual results are highly unlikely to match the ideal ratio. Now we're looking at the two characters, also a test cross. So these test cross is not limited only for one character, again for two characters. It's either individuals who are expressing two dominant traits, but the genotypes are unknown. So for example, the expression of the yellow round seed phenotype in the F2 generation, which is described, okay, resulting from this particular genotypes. The big G, homozygous G and homozygous W, homozygous G and heterozygous W, heterozygous G and homozygous W, and lastly, heterozygous G and W genotypes. So that's also applicable for particular test cross. And now, we're trying to solve this particular problem. So you want to try this at home. Considering the Mendelian traits round versus wrinkled and yellow versus green, consider the crosses below and determine the genotypes of the parental plants by analyzing the phenotypes of their offspring. So we have your parental plants, we have round and yellow, okay, both round and yellows, wrinkled yellow and round and yellow, round and yellow, and round and yellow and wrinkled and green. So these are their offsprings. So just to give you a hint, this problem will involve you a series of like Mendelian dihybrid crosses, and you are asked to determine the genotypes of the parents in a number of instances. So the parental genotypes are unknown, and the key to its solution is to write down everything that you know for certain. Okay, and it will reduce the complexity of this problem. Okay, it reduces also the problem to its bare essentials and clarifying what you need to determine. For example, okay, the wrinkled yellow plant in case, the wrinkled yellow plant here. So, your letter B must be homozygous for the recessive wrinkled alleles. Again, your letter B must be homozygous for the recessive wrinkled, because the wrinkled trait is tend to be the recessive, round is the dominant, okay, and it must bear at least one dominant allele for the yellow trait. So it's either B. Having established this, you will need, okay, or you need only to determine the remaining allele for the seed color. So that's technically getting all these combinations, these offsprings. You will be able to solve the problem. And now we're heading to the next portion. So now we're looking at the dihybrid crosses and demonstrating that Mendel's principle applied to inheritance of multiple traits. Your trihybrid cross thus far have considered inheritance of up to 2 pairs of contrasting traits, and Mendel demonstrated the processes of segregation and independent assortment also apply to 3 pairs of contrasting traits in what is called now as your trihybrid cross or 3-factor cross. Now, you'll see here a trihybrid gamete formation. Okay, this is theoretical contrasting traits which are symbolized by letters A, B, and C. In this cross between your homozygous ABC and homozygous small ABCs, all the F1 individuals are heterozygous for all the 3 gene pairs. So we got here a triplet of these gametes, okay, uppercase ABC and lowercase ABC. So if you try to get all the F1, you'll get all heterozygous. Now, when we try to cross all of them, okay, we got 8 possible gametes. So for us to better understand this some techniques that we're using in genetics, we have what we call as the forked line method or the branch diagram, because it is much less difficult to consider that each contrasting pair of traits separately and then to combine these results, okay? And we use this forked line method. This method also is what we call as a branch diagram, because it relies on the simple application of the laws of probability, which is established from the previous cross. We call that the hybrid cross, and each gene pair is assumed to behave independently during your gamete formation. So this generation of your F2 trihybrid phenotypes, we got it's either a small a or big A, small b or big B, small c or big C. Now we got this, since this big A is dominant, it will be going into the idea of 3:1 ratio for monohybrid crosses. 3:1, 3/4, 1/4, getting a big B and small b, getting a small c or big C, and we try to multiply all of them to get this particular proportion and all of these genotypes. So this is what we call as a forked line method. It resembles like a fork. Okay, this method is like based on your expected probability of occurrence for each of the phenotype. So we have, like, let's say for example, we got this homozygous ABC. Okay, there is a 27/64 chances that we got this genotype from the offspring. So among all of these, among 64 individuals from this trihybrid cross. Okay, let's move on with the next part. Now we are looking at Mendel's work, which was rediscovered in the early 20th century. Okay, Mendel's work was technically published in 1866, and his findings was often cited in every discussion, and it's being talked about in his place. Their significance were somehow like unappreciated for about 35 years. Then, in the latter part of the 19th century, a very remarkable event, okay, was held in recognition of his work. Walter Fleming's discovery of chromosomes in the nuclei of the salamander cells. Year 1879, Fleming described the behavior of these thread-like structures when he tried to observe these salamander cells during their cell division process. Okay? As a result of Fleming's findings, and the work of many cytologists, the presence of discrete units within the nucleus soon became the integral part of the scientist's knowledge or ideas about inheritance. This is Walter Fleming and his illustrations of the cells with chromosomes in the mitosis phase. This is from this particular book. And the polythene chromosomes in Chironomus salivary gland cell, which is one of over 100 drawings from the book, Fleming's book. And also, these are the people: Hugo de Vries, Karl Correns, Austrian botanist Erich Termach was founded the principles of Mendel and tried to improve something from that idea. Boveri Sutton, okay, also was being discussed in the introductory to genetics part. Again, they were the first one to formulate the chromosome theory of inheritance, and stating that inherited traits are somehow controlled by genes, or Mendel was telling us the unit factors. Okay, they are faithfully transmitted through gametes, and maintaining the continuity of these number from generation to generation. These are the 20th century's geneticists. Now we're looking at unit factors, genes, and homologous chromosomes. This is an illustrated correlation between the Mendelian postulates. The first one is your unit factors in pairs. Okay, this is having a homozygous G homozygous small g, w, and small w. This letter A. Letter B is your segregation during the anaphase. Okay, the first meiotic anaphase. These unit factors are segregating, okay, towards the opposite poles of your cell. They are being pulled out by your microtubules. As you notice, each pair is separating. It's either this way, okay. This big G and big W will go in one cell. The other one is small g or small w going to other cell. It could either be vice versa, as you notice on this second image. Okay, and lastly, we have independent assortment, which showing the presence of genes located in homologous chromosomes and their behavior during meiosis. This one. This is your independent assortment. So these meiotic products, okay, were not influenced by any other factors, any processes. These are possible gametic combinations which are formed with equal probability. So to illustrate, okay, this principle of independent assortment, it is very important for us to know or distinguish between members of any given homologous pair of chromosomes. One member of the pair, like one member could be either red or could either be blue. Okay, so that's why we are representing these different colors, like red for maternal and blue for paternal. And following this independent segregation or assortment of each of the pair of these homologs, each gamete receives one member from each of the pair of the chromosomes, and they must be equal. And for the next lesson, we now have the independent assortment, which leads to a very extensive genetic variation. So these possible genetic variation is not only limited to animals, not limited to plants, fungi, bacteria, and every living organisms on Earth. We can see it on our faces, it's on our fingerprints, morphological appearance, okay, even the color of our eyes. All of these will lead us to genetic variation, and thus leading, okay, for a very extensive biodiversity among all of these physical traits. So we have seen that the number of possible gametes, each with different chromosome composition, is 2n. Okay, 2, which is raised to the power of n. N is equals to the haploid number. Thus, if a species has a haploid number of 4, okay, then 2 is to 4. It could be 16. And there are 16 different gamete combinations which can be formed as a result of independent assortment. Now, although this number is not high, consider the human species. Okay, our haploid number is 23. Now, we compute 2 is to 23, and we got an excess of 8 times 10 to the power of 6, and that's over 8 million. So these different types of gametes are possible through independent assortment. Take note of it. It's 8 million different types of gametes. Okay? And one of the consequences of independent assortment is the production of an individual of like genetically dissimilar gametes. And since genetic variation is resulting of 2 members of any homologous pair of chromosomes, if ever these individuals are homologous, okay, very rarely that they are being genetically identical. Okay, as because you notice the maternal has a factor, the paternal part is a factor, and these factors will contribute or distributed to the gametes through independent assortment, and they will not be following a certain guidelines or for particular rules or mechanisms that will just you belong here in this gamete, you belong here in this gamete, and all of these possible chromosome combinations are produced, which is leading to a very extensive genetic diversity, and that's very happy for that particular phenomena. this is the fertilization event. Okay, fertilization, very as a breakthrough for mammalian species. Okay, this is in the laboratory. Only one of approximately 8 times 106 possible gametes from each of two parents. Okay, like you multiply that one 8 to 106, and we got 2 or 1 of only 64 times 1012 potential genetic combinations. Given that this very big probability, it's less like 1 trillion, and it's no wonder that except for identical twins, each member of our human species has this distinctive set of traits coming from a hair color, eye color, skin color, the stature, the posture, the combination, intelligence. Okay, very distinctive set of traits, and this number of combinations of chromosomes is far greater than the number of humans living on Earth. So we'll now try to look at the computational part of Mendelian genetics. We now try to look at the laws of probability, okay, helping us to explain genetic events. Probability, as you all know, a branch of mathematics which is concerned with numerical descriptions of how likely an event is happening or to occur, and how likely it is that proposition is true. Okay, and the probability of an event is a number between 0 and 1. Roughly speaking, 0 indicates impossibility and 1 indicates certainty. Try to look at genetic ratios. Recall that genetic ratios, for instance, your 3/4 tall and 1/4 dwarf, are most properly thought of as probabilities. And these values predict the outcome of each fertilization event, such that the probability of each zygote having genetic potential for becoming tall is 3/4. Okay, whereas the potential of its being a dwarf is only 1/4. Probabilities again will range from 0, it's certain not to it's very impossible to occur, or 1, which is very certain to occur. You will see here a number of different color of dice. Okay, now we're looking at the product law. When there are 2 or more events with known probabilities that are occurring or happening independently but at the same time, we are able to calculate the probability of their possible outcomes that are occurring together. So your product law states that the probability of 2 or more independent events occurring simultaneously is equal to the product of their individual probabilities. And we take a look at these coins, okay, coin activity, to illustrate this possible probabilities using your product law. We consider the possible results. So if you toss 1 peso coin and a 5 peso coin, which is denoted by P and N, at the same time, then we are able to examine all combinations of heads and tails that can occur. There are 4 possible outcomes. For the first part, we have here possible 1 peso coin head and 5 peso coin head, and then there is a half multiplied by half probabilities, then we get 1/4. Because this is a product law, you just have to multiply the probabilities of each of the 1 peso coin and your 5 peso coin and their probability of getting a head. Okay, so we also have this probability of getting a tail for 1 peso coin and a head for 5 peso coin. Then you get have 1/2 times 1/2, and you get 1/4. The same thing goes with your head of 1 peso coin and tail of your 5 peso coin, and lastly the tails of both your 1 peso coin and 5 peso coin. So if the photo on your left is a 5 peso coin. Next is your sum law. If we want to calculate the probability when the possible outcomes of 2 events are independent of one another, but it can be accomplished in more than one way, we can further apply your sum law. For example, what is the probability of tossing our 1 peso coin and 5 peso coin and obtaining 1 head and 1 tail? So the sum law states that the probability of obtaining any single outcome where that outcome could be achieved by 2 or more events is equal to the sum of the individual probabilities of all such events. Thus, according to your sum law, the overall probability in our example is 1/4 plus 1/4. There we get 1/2. In this example, there is independence. There is an independence between 1 peso coin and 5 peso coin in obtaining 1 head or 1 tail. It could either be 1 head for 1 peso coin or 1 head for 5 peso coin. It could either be 1 tail for 1 peso coin or a tail for a 5 peso coin. Thus, you just have to add all the overall probabilities. 1/4 plus 1/4 is equals to 1/2. Next, we're moving on to activity. We have your chi-square analysis, which is evaluating the influence of chances on genetic data. Mendel's 3:1 monohybrid ratio and 9:3:3:1 dihybrid ratios are hypothetical predictions. These are based on the following assumption. Number 1, each allele is either dominant or recessive. Number 2, segregation is unimpeded. 3, independent assortment is occurring. And lastly, fertilization is random. Now, as we're trying to look at the chi-square analysis that evaluates the influence of chances on our genetic data, let's understand first what are chance deviations and what does this concept attribute to our chi-square analysis. So this one, we'll try to look at the final two assumptions. Okay, they are both influenced by chance events and therefore they are subject to random fluctuation. This concept of chance deviation is most easily illustrated by tossing a single coin numerous times and recording the number of heads and tails being observed. So as the total number of tosses is reducing, okay, the chance deviation increases. Okay. For example, if a coin is tossed only 4 times, you would not be too surprised if all of the 4 tosses resulted in only heads or only tails. For 1,000 tosses, however, 1,000 heads or 1,000 tails would be most unexpected. Okay? In fact, you might believe that such a result would be very impossible to happen. Actually, all heads or all tails in 1,000 tosses can be predicted to occur with a probability of 1/2, which is multiplied to 1,000. Since 1/2, okay, 20 is less than 1 in a million times, an event with a probability as small as 1/2 times 1,000 is like virtually impossible. So this is the concept of chance deviation. And there are 2 major points that we should keep in mind when predicting or analyzing genetic outcomes. These are, the first one is that the outcomes of independent assortment and fertilization like coin tossing are subject to random fluctuations from their predicted occurrences as a result of chance deviation. And as the sample size increases, the average deviation from the expected results also decreases. Therefore, a larger sample size diminishes the impact of chance deviation on the final outcome. And it's very true when you are gathering data. It's not always there is a threshold or a standard sample size that we are meeting in a research study for us to be able to get the average deviation, okay, and decrease their numbers or their values. Okay. So now we are looking at chi-square calculations and the null hypothesis. In genetics, being able to observe, okay, deviation is a very crucial skill. When we assume that the data will fit in a given ratio, such as the 1:1, 3:1, 9:3:1, we establish what we call as the null hypothesis, or it could be denoted by H subscript 0. It is so named because the hypothesis assumes that there is no real difference between measured values, or the ratio, and the predicted values. There's no real difference, and any apparent difference can be attributed purely to chances. And the validity of your null hypothesis of a given set of data is measured using statistical analysis. And depending on the results of this analysis, the null hypothesis could be either be rejected or fail to be rejected. If it is rejected, then the observed deviation from the expected result is judged, okay, not to be attributable to chance alone. In this case, the null hypothesis and the underlying assumptions leading to it must be, re-examined. You must re-examine it. And if your null hypothesis failed, okay, to be rejected, any observed deviations are solely attributed to chances. And this is how you're going to do it. Chi-square analysis. Okay, this test takes into account the observed deviation in each component of a ratio from what was expected, as well as the sample size, and reduces them to a single numerical value. So the value of x squared is then used to estimate how frequently the observed deviation can be expected to occur strictly as a result of chance. And the formula used in chi-square analysis is this. Okay, x squared equals the summation. Okay, the Greek letter sigma represent the sum of the calculated values for each category in this ratio. So O minus E is observed minus expected, okay, squared divided by expected. Thus, this equation is being reduced to this x squared, or the chi-squared, is equals to the summation of your deviation squared divided by your expected outcome. Okay, so this is the most simplest statistical test for assessing the goodness of fit, okay, of the null hypothesis, which is your chi-square analysis. And to make sure that you understand how each number was calculated in this sample, you need to take note of this diagram, okay, this table of monohybrid and dihybrid. So we have here expected ratio for the monohybrid. You can remember from Mendel's garden pea plants, we have 3/4, 3:1 ratio. Observe, we have 740 and 260 for the 1/4. Okay, just to multiply, we got these expected values from this 3/4, okay, multiplied to 1000, and we got 750. 1/4 we get multiplied to 1000, and we get 250. And then your deviation, simply the D, is the difference between your observed and your expected results. Then we have here -10 and +10. So the deviation is just simply the squared of your the deviation squared. So okay, you square it 100. Now we try to compute the chi-square by simply putting the quotient, okay, which is 0.13 for the 3/4 and then 0.40 for the 1/4 expected ratios. And we got this result, which is the chi-square is your 0.53. So this p-value, we'll be learning about it in the future slides. Same thing with the Dihybrid Cross, since we are expecting them to follow the fashion or the ratio of 9:3:1. We also have the observed numbers, okay, from this section, and the expected numbers coming from just simply multiplying, okay, 9/16 to 1000. and we got this x squared values of 4.16, and later we will try to discuss what the p-values are for. So the final step in your chi-square analysis is to interpret your x squared value, and to do so you must initially determine a value called the degrees of freedom, which is n minus 1. Okay, n is the number of different categories into which the data are divided. In other words, the number of possible outcomes. For example, 3:1 ratio. Okay, your n is equals to 2. Go. So there are n is equals to 2. So you minus it, you deduce 1, then you get a degrees of freedom of 1. For the n is a 3, for the 9 is a 3:1 ratio, you got n is equals to 4. Okay, and the degrees of freedom is then 3, because 4 minus 1 equals to 3. You must take into account this degrees of freedom, okay, because the greater the number of categories, the more deviation is expected as a result of chances. Okay, so more expectations, okay, of chances in a dihybrid cross as compared to a monohybrid cross. So these what do you call this one? Your different categories will just refer to the possible, okay, phenotypes of the cross between organisms. And once you have determined the degrees of freedom, you can now interpret the x squared value in terms of the corresponding probability value, which is your p. Since this calculation is complex, we usually take the p-value from a standard table or graph. So using the graph, we need to execute the following steps. First, locate the x squared value on the abscissa. It's either the horizontal axis or the x-axis. Next, draw a vertical line from this point up to the line on the graph representing the appropriate df. So again, we'll try to look at the abscissa, the x-axis, and then a vertical line from this point to the line representing the appropriate degrees of freedom. From there, extend a horizontal line to the left until it intersects the ordinate or the vertical axis or the y-axis, and then estimate by interpolation your corresponding p-value. So this is your graph on how we convert chi-square values to p-values. Try to look at this. X squared values, we have here a 0.53, so this is 0.01 here, 0.553, and we take a look at your left, which is p48. Okay, since this one, this shade here in your graph, very wide. Okay, this figure is showing you a wide range of x squared values and the corresponding p-values. This is your x-axis, this is your y-axis. Okay, for the degrees of freedom in both a graph and also using a table. And let's use the graph to explain how to determine the p-value. So your graph for converting x squared values to p-values, and this letter B is a table for the x squared values for selected values of df, or the degrees of freedom. So 1 until 50. Okay. Values leading to a p-value of less than 0.05. So this one is your p-value. Okay, p-value of less than 0.05. So this is the lighter blue areas. This one, 0.05, lighter blue areas. Justify rejecting, okay, your null hypothesis. For example, the table in part B. Okay, this table in part B shows that your x squared of 0.53, your chi-square value of 0.53, okay, with 1 degrees of freedom, this one, 1.51, this here, okay, and the corresponding p-value is between 0.20 to 0.50, because this your 0.53 is included in that particular range. Okay, and the graph in A will give you more precise p-value, okay, let's say your p for your 0.53 chi-square value will give you a this range of your probability or your 0.48. Then using this interpolation, thus we fail to reject your null hypothesis. Now we'll try to interpret the probability values. Okay, and understanding the meaning of the p-value is very critical, because it's very simple to think of the p-value as a percentage. And let's use the example of the dihybrid cross in the previous table, where we got p is equals to 0.26, which we can thought of as 26%. Okay, and in our example, the p-value indicates that if we repeat the same experiment many times, 26% of the trials would be expected to exhibit chance deviation as great or greater than that seen in the initial trial. So that is conversely 74%, because 26 plus 74 is 100, and 74% of the repeats will show less deviation than the initially observed, which technically it is a result of chance. Thus the p-value, okay, reveals that your null hypothesis concerning, because it's a dihybrid cross, it's 9 to 9331 ratio, is never proved or disproved absolutely. Instead, a relative standard is being set, okay, that we always use to either reject or fail to reject your null hypothesis, and that standard is the p-value of 0.05. Okay, when applied to chi-square analysis, your p-value less than 0.05 means that the observed deviation in the set of results will be obtained by chance alone, less than 5% of the time. And such a p-value indicates that the difference between observed and predicted results is substantial and requires us to somehow reject your null hypothesis. P-values of 0.05 or greater, like 0.05 to 1, indicate that the observed deviation will be obtained by chance alone 5% or more of the time. And this conclusion will allow us not to reject your null hypothesis, because we are always using your 0.05 as our standard. Thus, its p-value of 0.26, the null hypothesis that independent assortment accounts for the results, fails to be rejected. So we accept. Okay? Therefore, the observed deviation can be reasonably attributed to chances. And a final note is very relevant here concerning the cases where the null hypothesis is rejected. That is where p is less than or equal to 0.05. Suppose that we had tested a data set to assess the possible 9:3:3:1 ratio, but we rejected the null hypothesis based on our calculation. So what are the alternatives interpretations of this data? Researchers will reassess the assumptions that underlie your null hypothesis. In our dihybrid cross, we assumed that, again, segregation operates faithfully for both gene pairs, and we also assume that fertilization is random and that the viability of all gametes is equal regardless of genotype, and that is, the gametes are equally likely to participate in fertilization. And finally, we assume that following fertilization, all the pre-adult stages and adult offsprings are equally viable regardless of their genotype. And if any of these assumptions is incorrect, then the original hypothesis is not necessarily invalid. Okay, we're moving on to the next topic. We are in creating pedigrees. Pedigrees reveal patterns of inheritance of human traits. Pedigree, such as a family tree, is called a pedigree. By analyzing a pedigree, we may be able to predict how the trait study is inherited. For example, is it due to a dominant or a recessive alleles? And when many pedigrees for the same trait are being studied, we often ascertain the mode of inheritance. Okay, this is a very traditional way to study the inheritance in order for us to construct a family tree. It indicates the presence or absence of the trait in question for each member of the generation. So the photo on the title slide will reveal you a very old pedigree, okay, from a royal family. So these are the following symbols, or we call them conventions, that we're constantly using in a pedigree. We have here male, which is designated by squares; female, circles; sex unknown is diamond. Okay, affected individuals are shaded. Okay, unrelated parents, consanguineous parents, there are two dashes; offsprings in birth orders, and we enumerate them; fraternal twins, which their sex may be same or different. These are identical monozygotic twins. The sex must be the same. In this case, they're both females. Multiple individuals are unaffected, so we put numbers inside. Proband, in this case a male, we try to examine that one. Deceased individual, in this case a female. Heterozygous carriers, they have the trait within them, but it's not visible or being expressed in the phenotypes. And these are your successive generations. So twins are like indicated by these diagonal lines, which are stemming from a vertical line here to a sib-shaped line. For identical or monozygotic twins, we have horizontal line, okay, this horizontal in between them. Fraternal lack this connecting line. A number within one of the symbols represents the number of sibs or siblings of the same sex and of the same or unknown phenotypes. The individual's phenotype, which is first brought to attention, was this, what we call as your proband, which is indicated by an arrow, okay, connected to a designation which is P. This term applies for either a female or male. So pedigree analysis, okay, which is the traditional way, okay, of studying the inheritance of a certain family members. It has always been useful for constructing a family tree, okay, and the diseases that were present in a certain clan or family. The first is what we're trying to do now is to look for different cases. The first is a representative pedigree for a trait which somehow demonstrates in autosomal recessive inheritance. We call it albinism, where the synthesis of the pigment melanin is somehow obstructed. Okay, individuals weren't your melanocytes weren't able to produce the melanin which protects your skin from the UV rays coming from the sun. So make sure, okay, that you are able to understand these pedigrees. So either 1, 1-3 or 1-4 must be heterozygous. Okay, in this example, you'll also see that it's skipping generations, and recessive autosomal traits are appearing equally in both sexes. So this is autosomal because it's affecting your autosomes, okay, your somatic cells. Recessive, okay, recessive alleles, okay. In this case, there is a proband, which is this 3-4 individual from this pedigree. We also have in next example, and our second pedigree is illustrating the pattern of inheritance for a trait we call it Huntington disease, which is caused by an autosomal dominant allele. So the key to identifying a pedigree that reflecting a dominant trait is that all affected offspring will have a parent that also expresses the trait. It is also possible by chance that none of the offspring will inherit the dominant alleles. If so, the trait will cease to exist in the future generations. So in this example, okay, we have here 1-1 is heterozygous for the dominant allele. So the first step to doing this is you will assign the labels, okay. For this autosomal trait, assign your genotypes. For this second generation and third generation, in this autosomal dominant trait, okay, it it appears in every generation. The 3 generations it appeared for males and females, and affected individuals all have has an affected parent. This one is their dad. This son, okay, will transmit it to his daughter and son. This daughter from the first generation transmitted the trait into her son. Okay, so dominant autosomal traits appear equally in both sexes. If that happens on only males or only females, that is what we call as your sex-linked traits, recessive or dominant traits. Okay, so there's good representative of recessive and dominant human traits. So as I say, pedigree analysis of many traits has been extremely historically valuable research techniques in human genetic studies. However, the approach does not usually provide the certainty of the conclusions obtained through experimental crosses, because it yields large numbers of offsprings. Nevertheless, when many independent pedigrees of the same trait or disorder are being analyzed, there is a consistent conclusions that could often be drawn, and it is showing us here in this table the recessive and dominant traits, and classifying it's either recessive or dominant. We have here albinism, alkaptonuria, color blindness, cystic fibrosis, Duchenne muscular dystrophy, galactosemia, hemophilia, Lesch-Nyhan syndrome, phenylketonuria, sickle cell anemia, and Tay-Sachs disease. And dominant traits, we have here achondroplasia, okay, brachydactyly, Ehlers-Danlos syndrome, hypotrichosis, Huntington disease, hypercholesteremia, Marfan syndrome, myotonic dystrophy, neurofibromatosis, phenylthiocarbamide testing, and porphyria, or some forms. Often, these traits, or sometimes are they are being tested, okay, for example in newborn screening. In the Philippines, we examine this. The DOH is examining these congenital genetic defects, for example galactosemia, okay, phenylketonuria, Tay-Sachs disease, because these are diseases that should be diagnosed in the very earliest stages of these newborns. And often they got the samples from the blood samples from the feet of your infants. Okay, so there's an activity now for this pedigree for myopia or nearsightedness, okay, in humans. You predict whether the disorder is inherited as the result of dominant or recessive trait, and determine the most probable genotype for each individual based on your prediction. So this problem is involving a series of Mendelian dihybrid crosses, okay, where you asked to determine the genotypes of the parents, okay, and in a number of instances, they're the family members. The key to its solution is to write down everything that you know for certain. Okay, this reduces the problem to its bare credentials and clarifying what you need to determine. For example, you try to locate, okay, to assign genotypes for this particular problem, and now you're able to see that these affected individuals, it's either as a result of a dominant or recessive traits. And lastly, we have here important topics, okay, as we end this lesson for Mendelian genetics. We have mutant phenotypes, which have been examined at the molecular level. So only recently, over 100 years after Mendel has, used his garden pea plants for his experimentation, okay, very groundbreaking when these wrinkled, okay, peas have come to find out how this wrinkled gene is able to make the peas being wrinkled, okay. So the wild type of these the wild type allele of this gene, okay, encodes a protein. We called it the SBEI, or the starch branching enzyme. This enzyme catalyzes the formation of the wrinkles, okay, of this seeds of the pea plants, okay. Um, as a consequence of this mutant form, okay, of this SBEI, because once there is a mutant form, there is a from lack of activity of this particular enzyme in pea plants, and the production of branch points is inhibited during the synthesis of starch within the seed, which in turn leads to somehow an accumulation or clogging of more sucrose in a higher water content while the seed is developing. And due to osmotic pressure, okay, inside the seed rises, causing the seed to lose more water, ultimately resulting in a wrinkled appearance in maturity. So this is the molecular explanation why these garden peas are becoming wrinkled. And in contrast, developing seeds that bear at least one copy of the normal gene, it's being either homozygous or heterozygous for your dominant allele, synthesize starch and achieve an osmotic balance that minimizes the loss of water. The end result for them is somehow a smooth, textured outer coating, so the round coats. And how this is being done in the lab, okay, it is being examined by cloning and analysis of your SBEI gene, which has provided us with new insights into the relationships between the genotypes and the phenotypes. And interestingly, the mutant gene contains a foreign sequence of somehow 800 base pairs that disrupts your normal coding sequence. And this foreign element closely resembles sequences that is being called as your transposons or transposable elements, or we often call that one as jumping genes. Okay. They have been discovered to have the ability to move from place to place in the genome of certain organisms, and transposable elements have been found in maize, parsley, snapdragons, and fruit flies, among many other organisms. Very interestingly, this person on your right is Barbara McClintock. He discovered transposition or transposable elements, which result in the duplication of the genetic materials. And her discovery of these transposons earned her a Nobel Prize in 1983. Amazing. Now, lastly, we are trying to look at the Tay-Sachs disease, which is a molecular basis of a recessive disorder in humans. Very important and very interesting cases where a single mutant gene, okay, causes multiple effects associated with a severe disorder in humans. And let's consider this modern explanation of the gene that causes Tay-Sachs disease, or your TSD. Tay-Sachs disease is a recessive disorder involving an unalterable destruction of your CNS. Okay? Infants with TSD are unaffected at birth and appear to develop normally until they are about 6 months old, and then a progressive loss of mental and physical abilities occurs, and afflicted infants eventually become blind, deaf, mentally retarded, and become paralyzed, and often within a year or two, seldom living beyond age of 5. And typical of rare autosomal recessive disorders, two unaffected heterozygous parents who most often have no family history of the disorder have probability in 1 in 4 of having a Tay-Sachs child. And the protein involved in TSD has been identified, and we have now a very clear understanding of how these underlying molecular basis of this disorder, and it is resulting from the loss of activity of your single enzyme hexosaminidase A, or hex A, which is normally found in your lysosomes. And remember, they digest waste or cellular waste or products within a cell. and it's needed to break down the ganglioside GM2, which is a lipid component of your nerve cell membranes. And without this functional hex A, gangliosides accumulates within the neurons in the brain and causes deterioration of your nervous system or central nervous system. This is very critical because heterozygotes of TSD with one normal copy of the gene, okay, produces about 50% of the normal amount of hex A. So heterozygotes of TSD are producing normal amounts of hex A, and they show no symptoms of the disorder. And the observation that the activity of only one gene, okay, is sufficient for the normal development of and function of your nervous system, okay, and somehow it explains the molecular basis of recessive mutations. And only when both genes are being disrupted by this mutation, okay, is the manifestations will be showing into the patients, or the TSD is showing to the child. Thus, the responsible gene is located on chromosome 15, and it codes for the alpha subunit of your hex A enzyme. And more than 50 different mutations within the gene have been identified that led to your TSD phenotypes. And that's the end of my presentation. It's all about the Mendelian genetics. I hope that you've learned something today, and thank you so much for listening. Don't forget to click like and subscribe to my YouTube channel. Thank you so much, and goodbye.